Okay, so let's start. Good evening, everyone. Welcome to today's asset colloquium, which is going to be given by Professor Areejit Samal from Institute of Mathematical Sciences, Chennai. For those of you who are from outside CIFR, I want to take a minute or two to introduce Asset Colloquium. Asset stands for Advances in Science, Engineering and Technology. It organizes colloquia at all working Fridays at 4, like the way we are having it here. Of course, due to COVID, we are having most of the talks online, especially the talks which are given by speakers from outside TIFR. So the asset usually organizes talks related, as I said, to engineering and technology. And of course, today's talk definitely comes with topics related to medicine and drug development and so on. And I'm sure seeing the title, it must be of very interest to the most of the people who are here on the platform. The title of the Phytochemical Atlas to Harness Traditional Indian Knowledge. I would like to thank Professor Vijay Gathar, who is also incidentally Rajaramana Fellow at IMSc Chennai, who actually proposed this talk. So I may request him to formally introduce today's speaker, Professor Datar.
Can you hear me, Samal?
Yeah.
Okay. Thank you, Dr. Satyanarayana. It's a pleasure to introduce Professor Areejit Samal. He's a faculty member, of course, as was said by. It's a pleasure. Pardon? Can you hear me?
Yes.
Yeah, okay. He's a faculty member in the Computational Biology Group of the Institute of Mathematical Sciences, Chennai, and head of the Max Planck Partner Group for Mathematical Biology. He was also a Ramanujan Fellow of the SERB and Simon's Associate of the ICTP until 2021. He did most of his studies, like bachelor's, master's, and PhD in physics. And I also learned that his thesis advisor was Professor Sanjay Jain, who of course, is also known for his work in string theory and so on. So after that, then he moved to several postdocs. For instance, he did a postdoc at the Max Planck Institute for Mathematics and the Sciences, Leipzig, 2008-2012, then the CNRS at Orsay in France, 2010-12, Institute for Systems Biology, Seattle, 12-13, and ICTP Trieste between 13 and 14. Then before he returned to India. His primary research interests are in the application of network science to study biological systems. And he has been responsible for creating one of the world's largest digital databases on Indian medicinal plants called IMPACT, IMP-PAT, with almost 30,000 entries. This will be of great importance to the discovery of new drugs by Indian companies, which has not yet happened, but hopefully it will happen. He is presently working on a database of endocrine disrupting chemicals and also on the systems modeling of metabolism and protein secretion system in microbes, apart from his core interest in the analysis of complex networks. So with this brief introduction, may I request Professor Areejit Samal to deliver his asset colloquium.
Thank you. Am I audible?
Yes.
Yes, yes.
I'd like to start by thanking Professor Datar and also Professor Satyanarayana for this kind invitation and giving me the opportunity to present our work. I'll start with a disclaimer. I'm neither a botanist nor a chemist. So this work and the different pieces of work I will try to present today was purely accidental, sort of foray into areas which got me interested. This was certainly not the plan when I moved back to India, but for the last 5 years, we've been working on this. And in general, area which I call as compilation, curation, and exploration of different chemical spaces. I'll start by introducing the work on the phytochemical space, which we basically have been working on for about 5 years now. But in general, I would like to, towards the end of the talk, mention the core area that we are developing of sort of computational analysis of these chemical spaces. So, use of medicinal plants to treat human elements is as old as human civilization. As you know, even the Greeks, the Egyptians, the Indian, the Chinese, all civilizations, if you look at an old text, medicinal plants have been documented which have been used to treat different human elements. What we need to recognize is that these plants basically use phytochemicals as chemical defense mechanisms against their competitors in the environment. And plant extracts containing these phytochemicals have been used to treat human elements for centuries. So as I mentioned, for example, in the Greek civilization, Hippocrates had documented more than 300 medicinal plants to treat different elements. What is important is that we should recognize that many present-day drugs were initially isolated from plants. So two prominent examples are the anti-malarial drug artemisinin and the anti-cancer drug paclitaxel, which are currently in use. More importantly, various systems of traditional medicine, including Indian and Chinese, use medicinal plants in their formulations. Moving specifically to traditional Indian medicine, in various old texts, for example, for Ayurveda, if you look at Charak Samhita, Atharveda, and other texts, there are at least 700 Indian medicinal plants documented in those texts. The traditional Indian medicine uses essentially several plants in their formulations, but such information, including the information on the phytochemical content, which is of importance to us, is largely buried within books. So it is largely in a non-digital format. And I would argue that the non-digital nature of this information on the phytoconstituents of Indian medicinal plants limits its effective use towards modern drug discovery, in general, any research towards human well-being in general. So when we started doing the work, what we recognized is that in contrast to Indian herbs, more comprehensive digital databases are available for the Chinese medicine, traditional Chinese medicine. For example, the database TCMID and TCM-Mesh contain more than 10,000 phytochemicals, which are produced by different Chinese herbs. So what we decided to do was to sort of bring the digitization of the information on the phytochemical content of Indian medicinal plants at par with what is available currently in the open space for traditional Chinese medicine. In short, what we have achieved is a resource called IMPACT. IMPACT stands for Indian Medicinal Plants, Phytochemicals and Therapeutic Uses. And this is, to my knowledge, the largest database on the phytochemicals of Indian medicinal plants. The version which is published since 2018 basically spans information on more than 1,700 Indian medicinal plants, 9596 phytochemicals, and more than 1,100 therapeutic uses. And this is sort of a resource which we have designed to facilitate computational approaches towards traditional knowledge-based drug discovery. While I emphasize on drug discovery, in general, the information in this resource can be used also for wellness. For example, if people are sort of designing herbal supplements, the information in IMPACT could be very useful. IMPACT is openly accessible through any of these following links. So let me just walk you through quickly 2 years of effort of digitization and curating and creating this resource. So the first step towards creating IMPACT was to create a list of Indian medicinal plants. So while this may seem simple, the use of synonymous names for different medicinal plants across the literature makes it very difficult to map the phytochemical content which is reported in the literature to a given plant. So what we did was we first created a non-redundant list of Indian medicinal plants. And in our first attempt, we had about 5,000 Indian medicinal plants. And these were largely collected from information which are captured by an organization known as FRLHT in Bangalore, the Ministry of Ayush, and the CSIR organization CMAP in Lucknow. So the idea was to basically map the synonymous names to a given accepted name, and this led to a non-redundant list of 5,000 Indian medicinal plants. Once we had the list of medicinal plants, we went ahead and tried to capture the known information on the phytochemical composition of these Indian medicinal plants. And here we basically resorted to 3 different resources. the major resource was the books. So we essentially ended up digitizing about 50 books for the first round of creation of impact. Then we also did some natural language processing, where we basically mined through abstracts in PubMed. Essentially, the information which is contained in impact comes from more than 7,000 abstracts. And then we are not the first ones to think of creating a resource of this type, but there were two existing resources. One by my colleague Ganesh Bagler, who now happens to be at IIIT Delhi. He created a resource called Phytochemical, which captured information on only 5 Indian medicinal plants, which covered 963 phytochemicals. And there was a second resource, which is since been obsolete, by Polo et al., which captured information on 295 Ayurvedic Indian medicinal plants and covers more than 1800 phytochemicals. So one of the issues with the existing resources is the following. The names of the phytochemicals is quite synonymous in the literature. And further, even if the name of a chemical is reported, it is not assigned a structure, or it is not mapped to a unique chemical structure. So one of the things we did was we sort of decided to have a non-redundant database. Like we created the non-redundant list of Indian medicinal plants, we decided to also create a non-redundant list of phytochemicals. So in this attempt, we had to do a lot of manual curation. Since the chemical names, as I mentioned, are synonymous in the literature, we had to remove the redundancy. And we basically did that by mapping the chemicals to, sort of standard chemical identifiers in various databases like PubChem, Kiwi, and CAS. Although we got about 30,000 strings in the first round, which could map to some phytochemical, we were only able to map 15,000 common names for different chemicals or phytochemicals to uniquely to 9596 standard chemical identifiers. Of course, you may ask, what about the remaining 15,000? That's work in progress, and we hope to map those strings also uniquely to chemical identifiers in some way or the other. And that is sort of an untapped resource as well. So what we were able to achieve in the first round in impact is basically information on 1742 Indian medicinal plants involving 9596 phytochemicals. Again, I want to emphasize that we did no experiments. This is just information collected from published sources. And furthermore, this is not the most comprehensive resource. We are only capturing known information from the published literature. Since one further point to emphasize is that in our database, every phytochemical has a chemical structure or 2D chemical structure assigned to it. So in contrast, if you take phytochemical, although it lists 963 phytochemicals for 5 Indian medicinal plants, only 500 of them roughly have chemical structure assigned to them. The remaining, so about half of them have no chemical structure assigned to them. So in that sense, our resource is computable. So since we have the 2D structure for all the 9596 phytochemicals in impact, we could immediately compute various physicochemical properties, the drug likeness of each of the phytochemicals, predict their admit properties, and also predict human target proteins from stitch. All this information that we have gathered for each phytochemical in our resource is captured in impact and publicly available to everyone to browse.
Areejit, if I can interrupt you, there was a question from Alak, one of our colleagues. There is a question, probably it's contextual, so I thought I will interrupt and ask you. The question is the following: could you provide some definitions as to how medicinal plants are perhaps only a subset of phytochemical, just so that we understand, I think the rest of the talk. Sorry if you have covered earlier and I joined it. Yeah, you did join a little late. So is that possible to answer this question?
No, can you repeat the question, please?
Okay, he was saying, could you provide some definitions as to how medicinal plants are perhaps only a subset of phytochemicals?
No. So, okay. So maybe, so I should have gone slow. Sorry. So medicinal plant is a plant which, through experience of, let's say some tribes, we know if you use it in some formulation has medicinal value. That is, it can be used to treat some ailment. Okay. So, but every plant and every organism produces many chemicals. So if a plant is able to, or a plant part is able to, we can use it to cure a ailment, then the chemicals in that plant are responsible for, perhaps responsible for the cure. So the chemicals which are in the plants are known as phytochemicals, particularly secondary metabolites, which are of interest. So basically, the idea is to go from experience-based science to evidence-based science. So the point is that we may know that, let's say, a herb is able to treat, say, antimicrobial. But in modern science, we would like to isolate the compounds which are within the herb, which would be able to treat treat against the microbes. So, for example, as I mentioned here in this slide, there are these drugs which are now used, like the artemisinin, the active ingredient, which were isolated from these plants. So that is phytochemicals. So why we are interested in phytochemicals is that phytochemicals is the active ingredients, likely to be the active ingredients, which are curing the different ailments. Okay, is that clear?
Yeah, I hope so. And there is another question which also from Professor Vivek Datar. You can see what is, I think it's a short form ADMET, ADMET.
Yeah, so absorption, distribution, metabolism, toxicity. So these are properties which are, sort of important for how it gets distributed. So once you take a capsule which has some drug, then it gets distributed in the bloodstream, and so these properties are important for designing a drug. So it's not important for the talk per se. So these are something which may be desirable. So some things you can have like a capsule, but some things you take as injections. So because you want directly, the drug to go into the bloodstream, for example. So maybe the ADMET is not that important, but these are some important properties for while designing a drug. Did I answer?
Yeah,
sure.
Yeah, sure.
So again, I want to emphasize that we are looking at the chemical content or the phytochemical content of plants, because these phytochemicals are the key to the curing of the human ailments. So, so after gathering the information on the phytochemical content of the plants, we moved on and tried to capture the information on the various therapeutic uses these plants could be used for. And here, it is a contentious issue. So we did not just go ahead and mine any literature. And so the therapeutic uses for the plants that we have collected are from standard books, typically books which are prescribed by Ayush Ministry. Furthermore, the therapeutic uses which are mentioned in these books are again in a very synonymous form. So to make our resource interface with, Western medicine, we did a minor innovation that we mapped those therapeutic uses to standard terminologies or identifiers which are used in Western medicine. For example, disease ontology, MESH, and OMIM. And actually, in the subsequent update that we are coming up with, we actually have the ICD codes which are recognized by WHO. So if you go to a hospital today, in most hospitals in Chennai, when they will diagnose you with some symptom or a disease, they will also give the ICD code. So we actually map it to the ICD code in the upcoming database. So, so the information on the therapeutic uses was also captured from various standard books, which are, recommended by Ayush Ministry. Finally, a third level of information where we did not want to reinvent the wheel was also to capture the information on traditional medicinal formulations. So, what is a formulation? Since this may confuse the audience, the formulation is actually the pharmaceutical, sort of preparation where you have the active ingredient and other things. For example, you maybe when you see a drug, it could be a sort of a syrup or a capsule or a tablet. So, and the formulations which are there in traditional medicinal formulations may not be just using one plant or one plant part, but it could be used based on multiple plants. So there is actually a large-scale effort by CSIR, which has created this traditional knowledge digital library called the TKDL. And they claim to have, over the last 15 to 20 years, digitized more than 2.5 lakh traditional Indian medicinal formulations. So there's no way we are going to compete with them. and our emphasis is on the phytochemicals. So what we did was we only mapped 1200 of the openly accessible formulations which TKDL makes accessible to us to the different plants in our database. So you may ask why TKDL is not public. I don't have the answer, but only they make part of the data and very small part of the data, which is 1200 openly accessible formulations, which is open to the public. The whole data is accessible to various patent offices, copyright offices, and they share it with various organizations only. Here we don't want to reinvent the wheel, so we only took the mapping of the 1200 openly accessible formulations and mapped it to various plants in our database. So, what we have, since I'll try to sort of walk you through the different levels of information we have. We have the medicinal plants, about 1700 or 1700 plus medicinal plants, and their phytochemicals. Again, we don't have information on all phytochemicals produced by plant. We document whatever has been reported to date. So we have 1700 plants and 9500 phytochemicals. Every plant doesn't produce all of them, few of them only. Then for each plant, we also have information on their therapeutic uses and also information on if they are used in some traditional Indian medicinal formulation, and that is only limited to the 1200 openly accessible formulations within the TKDL. Since we have the level, we can compute all these properties which I mentioned, and this is all made available through the resource. Is there a question? Then I can take it.
No,
okay, good.
I don't see it.
Okay, so I mean, this is about 2 years of work to sort of build this largest resource. I'm just giving an overview of what we did, and most of the steps are actually less computational and very much manual because much of the information was not in any digital format. We actually had to copy the information from books into the computer by hand, and then curate it manually. Actually, there's no other way to do it. Actually, so the question emerges is that we have these 9596 phytochemicals in our resource. How is this useful? And so now in the next part of the talk, I switch from documentation, that is compilation, to actually exploration of this chemical space. So again, you have to believe me. So there are certain measures or some properties or metrics which have been designed, which are based on essentially physicochemical properties of various chemicals, which are used in very initial phases of drug discovery to identify which are the molecules which are potentially drug-like. And I'm not a chemist, as I mentioned. So I took a wisdom of crowds approach. We took 6 different drug-likeness metrics. So they have some names like traffic lights, Pfizer 3/75, GSK 4/400. So as you can see, some of them have been actually devised by the pharmaceutical companies. And we subjected each of our 9596 phytochemicals to these drug likeness metrics. And I asked a very simple question. We basically asked a simple question: how many of our 9596 phytochemicals are drug-like based on all the 6 different measures we took? It turns out that 960 of them pass all drug likeness measures. So they are likely to be drug-like. What is interesting is that 32 of the 960 are already FDA approved drugs, that is the US Food and Drug Administration approved drugs. What is furthermore interesting is that 369, that is approximately 40% of the chemicals which are drug-like, have significant structural similarity to FDA approved drugs. So now you may ask, how do I quantify structural similarity between 2 chemicals? So since this is a colloquium, I wanted to keep the details out. Given 2 structures of chemicals, there are certain distance measures which are there to figure out, or similarity measures to figure out if 2 chemicals are similar or not. And one of the most popular measures is known as Tanimoto coefficient. We use the Tanimoto coefficient to decide if 2 chemicals are sufficiently similar or not. And it turns out that 369 of the 960 phytochemicals, which are in our database, which are drug-like, have some similarity to any approved drug. So we can view this space of 960 drug-like phytochemicals as a network as well. So here, every node or a dot here is one of the 960 drug-like phytochemicals. And there is an edge which is connecting two phytochemicals if they are similar. So as I mentioned, there is a metric that we use, which is known as Tanimoto coefficient. This is kind of a distance between 0 and 1. Doesn't matter for the details. So if two chemicals are similar by greater than 0.5, equal to 0.5, we put an edge between them. Furthermore, there is a color code to this figure. The red nodes, so any of the red phytochemicals here or chemicals are such that they have at least similarity to one FDA approved drug to date. And the green nodes have no similarity. So what is to be noted here is that although we have this 960 drug-like phytochemicals in the within impact, these chemicals are not always similar to each other. So as you can see, there are some clusters here and there are several disconnected clusters, smaller clusters. So this kind of suggests that this space is chemically diverse. So when you are looking for drug discovery, you want to go for new chemical scaffolds. So because there is a problem of antimicrobial drug resistance and so on. So you are looking for new drug chemical scaffolds as chemical spaces. So here is a space which, as you can see, is quite disconnected. And further, if you see, for example, this cluster, all the chemicals are green. That is, these chemicals are not similar to any of the existing FDA approved drugs. So these could be very interesting from potential future drug discovery. So this is unpublished data and we are further analyzing, and this could have further implications for potential drug discovery. So one of the first results we get by analyzing the chemical space is that there is a significant drug-like subspace within the impaired phytochemicals. That is 10%. Further, 60% of this space has no similarity to existing drugs. So there is opportunity for future drug discovery. A second question, which of course the referee asked, we didn't want to do this because it's a lot of work, is that how does it compare to the space of phytochemicals associated with Chinese herbs? Again, this analysis is limited to whatever data is available on Chinese herbs. So there is the resource, as I mentioned, called TCM Mesh, which captures information on almost 10,000 phytochemicals coming from 6,235 Chinese herbs. Although they had not done the drug likeness analysis, we repeated our drug likeness analysis on the 10140 phytochemicals in that database. What we found is that about 10% again, to be precise, 972 phytochemicals were found to be drug-like in all the 6 measures that we evaluated. Further, 19 of the 972 phytochemicals were already approved drugs. What is more important to ask is that what is the overlap between the phytochemicals for Indian herbs which are in our database versus phytochemicals for Chinese herbs in TCM. Turns out the overlap is approximately 25%. Similarly, if you look at the overlap between the space which we determined to be potentially drug-like, that is 960 for impact and 972 for TCM mesh, again the overlap is roughly 25%. So this suggests that although there is commonalities in the phytochemical space between Indian herbs and Chinese herbs, which is expected, there is a large space which is unique to the Indian herbs and also Chinese herbs. And so this suggests that the phytochemical space for both Chinese and Indian herbs provide extensive opportunity for drug discovery. So this is the second result that we got by analyzing the phytochemical space of Indian herbs and Chinese herbs. So this is a sort of a technical analysis, but I'll try to sort of convey the core message to the audience here. So this analysis was motivated by a paper by Stuart Schreiber's group, who is the head of the Broad Institute's drug discovery program. So what they have done in this PNAS paper is that they have analyzed 3 different sets of chemicals. One, a set of 6,000 molecules, which are commercial compounds, which are routinely used in drug discovery. Then a second set of about 6,000 molecules, which are known as diversity-oriented synthesis compounds. These are generated by academics for, chemists for various purposes in drug discovery. And third, which is a set of natural products. So natural products are chemicals which are produced by some organisms, not limited to plants. Could be microbes, humans, other organisms as well. So what they did is that they actually defined two size-independent quantities to quantify these different molecules. One which they call as stereochemical complexity, second which is known as shape complexity or FSP3. So these are very simple measures to compute. The first, the stereochemical complexity basically computes the fraction of carbon atoms in a molecule which are stereogenic. So if there is a carbon atom which is stereogenic, then you can have a stereoisomer around that carbon atom. Second, which is the fraction of carbon atoms that are sp3 hybridized. So if you have two carbon atoms which are sp3, then around the bond you can have a rotation. But if there is a double bond sp2, you cannot have a rotation. So if you have more sp3 carbons, then you have more shape. So this is a bit technical, but I want to give you the most important result they got. So what they found is that they actually took these molecules, which are the commercial compounds, the diversity-oriented synthesis compounds, and the natural products, and they computed the distribution of these quantities, stereochemical complexity and the shape complexity. And what they found is that on average, if you take the mean or the median of any of these two quantities, the natural products, that is molecules which are produced by organisms in nature, they are likely to have higher stereochemical complexity and higher shape complexity in comparison to commercial products or diversity-oriented synthesis compounds. So in general, if you see, the natural products or chemicals in nature are likely to have more stereochemical complexity and shape complexity in comparison to synthetic compounds. What they did further is amazing. See, they didn't stop here, but they actually went ahead and did some experiments, quite a bit of detailed experiments, where they took each of these molecules against a random assay of proteins. And they tried to quantify for each molecule how many proteins it could bind to. And what they find, which is very interesting, is that, and I will try to summarize in one sentence only, that in general or typically, natural products bind to very few proteins. So if you look at it, natural products bind to 1 or less than 5 proteins typically. But if you look at commercial compounds or diversity-oriented synthesis compounds, they bind to, typically to many more proteins. So why is this important? So if you are trying to design a drug, the drug has to go and bind to a certain protein or a drug target. And one of the issues with any drug is potential side effects. So if it binds to the desirable target, but also other off targets, it can lead to side effects. So you actually want a molecule which is very specific and goes and binds to the specific drug target and doesn't bind to other targets. So if you look at it, natural products are likely to be such molecules because they are going to be typically binding to one protein and not to other proteins. While if you have synthetic compounds, it may bind to many more drug targets. So this was amazing. So what they were able to do is basically correlate these two quantities, which are size-independent quantities for molecules, to this protein binding specificity. So what we could do is, certainly we cannot do the experiment, but we could compute the distribution of stereochemical complexity and shape complexity for the phytochemicals for Indian herbs and Chinese herbs. So I would like to mention that the set of natural products that took here, which is about 2,500, it had very little overlap with our phytochemical space. Actually, only 137 were common. So I basically take these histograms.
Areejit, I'm sorry, I missed a point. A question from Vasudev. Vasudev, can you please ask, because it was again a contextual for a particular slide?
It was on that slide where you showed this Basu of all the green and the red dots in some graph. And I was wondering what the X and Y axis on this space is. Like, how do I understand distance and direction in this space?
Basu, there is no X and Y axis here. So if each dot is a chemical.
But then why is, let's say, one cluster different from another cluster, and how do I understand how different it is on this plot?
So basically, if you take any molecule in one cluster versus another molecule in a second cluster, they have similarity less than 0.5 in this distance metric. This is just made visually to represent all the clusters. It has no specific value for this. But what is important here is that the red clusters, if you look at where the reds are, one point I wanted to emphasize is that there is some interest in finding sort of biosimilars. That is, can you find a drug which is a natural product, which is similar to an existing synthetic drug? So here the reds show that this cluster has many molecules which are similar to an existing FDA drug. So these could basically be used perhaps in place of the existing FDA approved drug. So, but there is no X and Y axis here.
Okay. Hey, on the same slide, there was a question from Atiya Jafar. It says in the same network, what does the size of the node signify?
Actually, the size of the node is just a distortion, unfortunately, because we are trying to show the different clusters, right? So it has been sort of tilted in an angle. So there is no relevance for the size of the node here, unfortunately.
Sure. Thank you.
So this is actually preliminary analysis. This is not published. So I'm sorry for the lack of quality here. So one of the key messages I wanted to say is that in this work by Stuart Schreiber, they are able to correlate these two quantities, which are shape complexity and stereochemical complexity, to the specificity of protein binding. Of course, we can't do the experiments for our phytochemicals, but we could check if the distribution of phytochemicals in our resource is similar to the natural product space, which is analyzed by Stuart Schreiber. So I basically replot these histograms as a box plot here. So I basically reproduce their data, the commercial compounds, the diversity-oriented synthesis compounds, and the natural products by Stuart Schreiber, and we basically compute the same stereochemical complexity distribution for phytochemicals of Indian herbs and for Chinese herbs. As you can see, it is very similar for the phytochemicals in terms of stereochemical complexity to the natural product set they took. And both for stereochemical complexity and shape complexity. Actually, in subsequent work, Stuart Schreiber has not just limited to these two measures, but 6 other physicochemical properties. And we find that in all physicochemical properties analyzed by Stuart Schreiber's group, the phytochemicals of Indian herbs and Chinese herbs are very similar to the natural product set. So what is the implication of this result? If I was allowed to extrapolate a bit, based on the work of Clemens et al., essentially the Stuart Schreiber paper, perhaps the impact phytochemicals are also likely to have higher fraction of specific binders to the target proteins relative to commercial compounds. Again, as I mentioned, when you are designing a drug, you want it to be very specific to its intended target and not have many off targets to limit the side effects. So essentially, what we have published in the first piece on impact is the following. So actually, we have 3 networks here, or 3 associations here. We have medicinal plants, which is 1742. We have their phytochemicals, which is the center stage of our work, 9596. We have also medicinal plants and their therapeutic uses, and some information on medicinal plants and their appearance in traditional Indian medicinal formulations. This was limited to the openly accessible TKDL data. And after creating the resource on the phytochemicals, we have done 3 different analyses. One, what is the space of drug-like phytochemicals? We show that there is a large space which is potentially drug-like, and further 60% of that space has no similarity to approved drugs. So there is opportunity for new drug discovery. We have compared our space with the phytochemicals of Chinese herbs and showed that the overlap is at best 25%. So both spaces have opportunity for new drug discovery. And also we have computed these physicochemical properties associated with specificity of binding to proteins, and we find that phytochemicals from medicinal plants are likely to be specific binders to human proteins. So if you look at it, this is where we are. So we have information of plant to phytochemicals, plant to perhaps their therapeutic effects. And what we would like in future is to link the phytochemicals to their target proteins explaining the therapeutic effect. So if the plant is actually curing a certain ailment, then the list of phytochemicals should be binding to certain important drug targets and explaining the therapeutic effect. And this is where we would like to go. And essentially, the long-term goal for impact is somehow to develop the platform based on computational biology tools to aid in traditional Indian knowledge-based modern drug discovery. So this was, of course, published in 2018 already. And we did it as really a side project, and given the attention and also the relevance we realized later on of the work, we have been working towards a large-scale update, which I think we would release in the next couple of months at best. So, of course, 1,700 plants is not all that we have in this biodiverse country. So, what we have been doing is we are essentially expanding impact, both in terms of the data and also in terms of the objectives. So we are coming up with a resource which will have now more than 4,000 plants. The number of phytochemicals has increased to more than 1,700. The phytoconstituents and therapeutic uses are now at the level of plant parts. So this is very important, because if you take a plant, you grow it in Himalayas versus if it manages to grow in Chennai, then the phytochemical content is going to differ. That is, even within different seasons, it differs. Further, if you look at traditional medicinal formulations, they use a specific plant part. That is, the phytochemical content of the root may differ from the leaf. So this level of information is quite available for some of the important plants. So now our updated resource would actually have both the phytochemical content and therapeutic use information at the level of these plant parts. And moreover, we have actually became aware of many important books. We have expanded the digitization to more than 70 books, and again further more than 7,000 articles. One of the things objectives is that to capture the structural diversity of the known phytochemical space, actually identifying new scaffolds, chemical scaffolds, novel chemical scaffolds, we will be able to do now. We are actually doing some drug repurposing analysis to identify phytochemicals similar to approved drugs. So to just give you an overview of what we are going to come up with. So the number of plants actually has increased twofold, phytochemicals about twofold. But if you look at the number of associations is actually fivefold now. And so this resource will be released very soon. Furthermore, you may ask if the Druglike space increased. So this is unpublished data. So we have now 70,967 phytochemicals. We have again computed the drug-like subspace. It is not 10%, but only 7.5% now, 1,335. But what is more interesting is that now 80% of these drug-like phytochemicals have no similarity to FDA-approved drugs. So what is more important is that we are finding new scaffolds here potentially. So this publication or manuscript should be communicated very soon. Of course, there is a lot of interest in using our phytochemical library for virtual screening for identifying new potential lead molecules. Essentially, during COVID times, even the Open Drug Discovery Hackathon, which was conducted by CSIR and PSA, they recommended IMPACT as one of the important libraries to screen, and we have also screened and found potential drug-like molecules for SARS-CoV-2 related drug targets here in 2 of the papers. So if you are still confused about why natural products, why phytochemicals, this is some unpublished data and a publication in work. So what we have tried to do in this work, and I'm trying to show you a graph here, is that we are trying to look at the number of molecules, small molecules, which are approved drugs since the inception of US FDA. So, FDA in its former avatar, the first drug it approved was morphine. But, in its current avatar, it came into existence around just after 1935, 38. So we actually got hold of all data from US FDA from their files. They actually gave it to us somehow. And we actually have this plot. If you see the brown plot, it is telling us the cumulative number of small molecule drugs which have been approved since 1938 to 2018. So we did it till 2018. We are expanding it now again. And what the green, graph is showing is the fraction of the drugs which are derived, which are either directly natural products or derived from natural products. That is, the drug was discovered with the core molecule being some natural product. So if you see, till 2018, about 2,800 molecules, small molecule drugs had been approved by US FDA, and almost 1,100 were natural product or natural product derived drugs. So natural products have contributed immensely to modern drug discovery. Lastly, just to mention, secondary metabolites of not just plants but also fungi have played very important role in drug discovery. So if you look at penicillin, one of the first important drugs, that is a fungal secondary metabolite. So actually in traditional medicine, not just medicinal plants, medicinal fungi, in particular mushrooms, have also been used to for centuries to treat ailments. So, and we have actually done a similar analysis, created a similar resource and analysis for fungal secondary metabolites for medicinal fungi. And I don't have time to summarize, but the analysis of the secondary metabolite space of fungi also shows that it has very similar properties to the phytochemical space of plants in terms of the stereochemical complexity, the shape complexity, the physicochemical properties, and there is a huge drug-like space. So this is another resource that we have created, and the exploration of the chemical space shows that there are parallels and similarities between the phytochemical space and the secondary metabolite space of medicinal fungi. So in general, the area that we have been developing in my group over the last 5 years is basically compilation, curation, and exploration of chemical spaces. And when we are talking of exploration of chemical spaces, we are taking both chemi-informatic approaches and network-based approaches. And the chemical spaces that we have been exploring can broadly be divided into two categories. One, which is potentially beneficial to humankind, and on this we have our resource impact, which is phytochemical space of Indian medicinal plants, and this resource called MEPSAT, which is secondary metabolites of medicinal fungi. But actually another area that we have contributed a lot, in my view, is spaces of chemicals, environmental chemicals, which are potentially harmful to humankind. And in this activity, we have basically released and analyzed different chemical spaces. And one particular class of chemicals which is of lot of current interest is the space of endocrine disrupting chemicals. So I will just take 5 minutes quickly to summarize some work which has actually been very nice, in the sense that we actually were able to contribute to the framing of a national policy in a Western country. So I'll just like to let you know about this for a bit also. But as I said, this is a general area that we are working on, both compiling, curating, and exploring the chemical space. It's not just limited to phytochemicals. So, this work that we have done on endocrine disruptors could be viewed from two areas of research which are upcoming. One, which is characterizing the human exposome. So exposome is sort of the environmental complement of the genome. And here there has been recently a call to use sort of network science approaches to characterizing the human exposome. So human exposure to environmental chemicals is an important contributor to disease burden, and there is immense interest in characterizing this external chemical component of human exposure. Essentially, if you look at it, in our daily life, we are exposed to various chemicals. For example, through pollution, we may be traveling every day for an hour and we are exposed to chemicals through pollution. So these have health effects on us, and this is now well documented. And there is a class of chemicals which has got a lot of attention, which are known as endocrine disrupting chemicals, in short EDCs, which are an emerging concern to human health. These, if you think of in, they are also termed in common terminology as hormone disrupting chemicals. So these are any plastics that you use, bisphenol A, these are all endocrine disruptors. Pesticides are endocrine disruptors. So there is a lot of interest in characterizing these endocrine disruptors, which are all over in our everyday life. Since I am running short of time, just to give you an overview, EDCs, they are defined by the World Health Organization as follows. An endocrine disruptor is an exogenous substance or mixture that alters the functions of the endocrine system and consequently causes adverse effect in an intact organism or its progeny or subpopulations. These chemicals actually happen to be omnipresent in our environment, including air, soil, and water. Some of these are actually from natural sources. So, for example, if you eat a lot of beans, beans has something known as genistein. It's a phytochemical. A lot of genistein can be an endocrine disruptor in your body. But most of these EDCs are synthetic chemicals which are used in human-made products. So these are man-made. Typically, they are in pesticides, cosmetics, and so on. And as I mentioned, there is a lot of interest in characterizing them and limiting their use in our daily life. So there is a large-scale effort, for example, in the European Union. So they have released this non-binding resolution in 2019 to come up with a comprehensive framework to identify endocrine disruptors in the European space. And in particular, France actually has a specific national policy called SNPE2, which stands for Second National French Strategy for Endocrine Disruptors for these chemicals. So I don't have time because I'm running short of time, but what I'll just quickly tell you is the following. Why is this important? And this actually has led to an emergence of a new field called computational systems toxicology. So if you look at toxicology, it is sort of a sister science for pharmacology. Toxicology studies basically adverse effects of chemicals on living organisms. The way traditionally toxicology has been done is that you basically test these chemicals on animals and characterize how, based on their adverse effect on animals, what they could cause to humans. But there is an ethical concern in using animals to test these chemicals and characterize their adverse effects. So there is a call, of course, for alternate methods. One other concern is that even if you limit just to the United States, because where there is large-scale labeling of products and what are the different chemicals in different products, there are more than 100,000, that is at least 1 lakh chemicals which are in commercial use. And only if you look at, even if you take the US Environmental Protection Agency, which has all these testing facilities, they have tested at best 10,000 of these chemicals today. So what about the remaining 90,000? We don't know what they are causing to us. So this has led to the emergence of a new field now called computational toxicology, which is an emerging area, which essentially tries to use mathematical modeling, very similar to what is used in computational systems biology, using even network biology to predict the adverse effect of chemicals on living organisms. That is, given the structure of a chemical, can we predict what its adverse effects would be through some computation? And in this regard, we want to basically build some predictive models for endocrine disruptors. I don't have time to go into this area in the work because I thought I would have more time. But what I want to just say is that when you want to build very good predictive mathematical or computational models, especially for EDCs, your model should have good data. If your starting data or the training data is trash, then your results and predictions are likely to be trash as well. So in that regard, we have actually produced a resource called DEDUCT, which stands for Database for Endocrine Disrupting Chemicals and the Toxicity Profiles. And this resource was created by 2 of my students. My first student who submitted recently, Janani Ravichandran, she actually went through 18,000 papers, actually 22,000 papers to date, and collected data from more than 4,000 papers to document in the current round, almost 800 chemicals which are EDCs with experimental evidence and literature. So what we wanted to tell you is that, of course, this is published. Again, we have not just created a resource, but we have done different analysis of the chemical space as well. And one of the important things that we learn here is as follows. So if you want to predict the effect of a chemical using some modeling, what is done routinely today is the following. Let's say we know the effect of some chemical A, and we don't know the effect of some chemical B. So what is routinely done in modeling today is that if A and B are similar, they would say that B would have an effect similar to A. This is what is known as QSAR, which is basically linear regression. But what we have shown by using the extensive data which is collected in our resource DEDUCT is that there is hardly any correlation between similarity in chemical structure to similarity in the targets and adverse effects. So this poses new challenges for predicting endocrine disruptors, and I don't have time to go into details, but we are thinking of using other systems biology approaches towards prediction of endocrine disrupting chemicals. What is very nice is that when we did this work, we didn't know how relevant it would be. So this is Ansys, which is the French Agency for Food, Environmental and Occupational Health and Safety, which is a French government organization. They are actually in charge of framing the policy SNPE2, which I mentioned, which is the second French national strategy for endocrine disruptors. Last year, they released a report, this is an 80-page English report, where they actually evaluated 27 worldwide initiatives on EDCs. So we were not the only ones. But after their evaluation and an 80-page document over 4 chapters, they decided to go with our resource for prioritization of chemicals for France. So this is some excerpts from the report. So they say that concerning the identification after reviewing the existing lists and methodologies, Ansys essentially recommends to deduct our resource for the clarity of the method used for identifying the substances and based on the criteria applied, selecting potential EDCs coherent with the food definition. So I didn't expect this, and this is really very heartening for the hard work of the 2 students who did all the work. With that, I'll just end because I went over time. This is some press coverage for both works that we have got, both on the phytochemicals and endocrine disruptors, and these are some people who really work. Most importantly, I would like to highlight the students who did the work. Vivek Anand Karthikeyan, who is now a PhD student in Dresden, and B.S. Karthikeyan, who's moved on to a postdoc in Sweden. They basically led the work on phytochemicals. Janani and B.S. Karthikeyan led the work on our exposome work and endocrine disruptors. Ajay Sahu, Kavya and Aparna contributed to various projects as well. And this is some of the collaborators I have. The work that I presented till recently has not been funded by any organization, but I have been fortunate to receive funding from various organizations. I thank them for the support. I'm sorry to have gone over time, but I'll be happy to take any questions now from the audience. Thank you very much.
Thank you very much, Areejit, for very, very nice. A lot of material that, of course, as you said, everything couldn't be covered in the given time, but it is obviously it's very, very interesting and very, I would say, far-reaching kind of applications and use for Indian pharma industry, drug industry, maybe any other beneficiaries as well. So there is one probably okay. There is sir. Before I take questions from Professor Datar, I would like to take question from Abhishek Sharma from NCBS Bangalore. Abhishek, you can ask yourself if you would like. Unmute yourself and ask the question. Yeah.
Hello, sir. This is Abhishek from NCBS Bangalore. Sir, I want to ask a question regarding that computational toxicology you were showing in one slide.
Yeah. Yes, please. This is the slide. Yes.
Yes. So, for example, now the FDA approved drugs are very less in number. For example, 2,000, and you have also shown that the chemical space, like the chemical similarity, has less to do with the effect which they are doing in the body. Okay. So now I want to ask, like, how you want to solve what's the rationale, how you want to solve this problem with computational system, as one cannot get the wet lab data on highly toxic drugs, so that we don't have any negative sets to compare.
Okay. So it was a bit patchy from your end. So if I understand correctly, what you're saying is that since chemical similarity is not correlating with target similarity, how do we become predictive? Is that the question?
Yes. And the second point to it is like we can't get the negative data set, like the effect of highly toxic substances, the wet lab data for the highly toxic substances. So in both regard, how will you solve this problem?
Yes, so you have hit the nail on the head. That's a fantastic observation you made. So even if you want to do machine learning to predict EDCs, what should be the negative data set, right? We can for sure say that a chemical is a EDC, but it is very difficult to say if a chemical is not an EDC. So it's not, I have been actually frankly pondering about that question a lot. What should be an ideal negative data set? Actually, if you have ideas, I would love it. Now you're asking how do we improve the predictions, right? So here in computational toxicology or whatever this terminology which people use, I mean, these are one of the things which is lacking is pathway-based modeling. That is, for the chemical, at least for some of the EDCs, at least in rat and mice, there is expression data available on their impact. Some data has been generated. Now, those type of pathway level details has not been incorporated. One other thing I didn't mention is that, I mean, since I had to go very fast, so there are actually three aspects of, let me just go here. So there were three goals actually I mean, that is why DUCT has been selected by the French organization. So the problem with the other initiatives on EDCs is that they mix data. That is, they compile information from fish studies, environmental modeling, monitoring, and computational predictions. But we decided to exclusively focus on evidence which is in vivo or in vitro in humans and rodents. So that sort of limits that space. Further, we have actually documented the adverse effects. That is, is it causing fertility problem or thyroid problem? So that level of information we have, whatever was captured in the papers. Further, we also have the dosage levels, which I don't think any resource has. That is, at which dosage different effects are observed. So this actually helps us in future to do much more. That is why DUCT is so sought after currently. I hope I have answered a bit. And the other thing which we need to think about is to sort of also capture the activity information. So, and that would help us model better and be better predictive. So this is an important challenge. I don't know how to do that. And further, there is one more important challenge is that we can perhaps predict the effect of a single chemical, but mixtures is not sure. This is both a challenge for the phytochemical work and also for the endocrine disruptor. In reality, we are encountering mixtures of chemicals rather than single chemicals. This has to be tackled. I'm sure. I hope I tried to answer.
Yeah, thank you.
Yeah, thank you, Abhishek. We have a question from Professor Druglike.
My question, Areejit, is regarding the possibility of finding drugs based on, let's say, the data that you have generated, which don't have, let's say, the side effects in diseases which are quite widespread. Let's say cancer. I mean, many of the cancer treatments have serious side effects, and as you say, if there are things that target only a specific aspect of the cell, maybe that will have less side effects. So is that a possibility or is that not a possibility for cancer?
Yes and no. So yes, in the sense the following thing that the first question to ask is that do we believe that a single chemical of the plant is responsible for the therapeutic effect? That is, the plant is producing many chemicals, right? And the cocktail is used to be, responsible for ailment. Okay. Let's say we first convince ourselves that the reported therapeutic effect actually exists. Then perhaps we can actually identify the phytochemicals or the major contributors to the sort of the therapeutic effect. I think it's a possibility. But what actually it requires is really experimentation and frankly, I have to expand my efforts to reach out to the chemists and people who really do the work, because this is largely computational till now.
Yeah, that's right.
That level actually. So I can tell you very simple ways of which our database can be used. For example, let's say you want to design a new drug against, say, some bacterial infection. Okay. We have, let's say, 10 plants. For the sake of discussion, we may have 10 plants in our database which are antibacterial. We can see what is common to the 10 plants, or we could see what could be unique to the 10 plants. Okay. that sort of analysis we have not yet managed to do, because we have been actually till now focusing on creating the largest digital resource, and actually we are in the phase now where we will turn from compiling to more exploration in the future. So this type of simple analysis will lead to several new hypotheses. But we have.
Can I just ask a very quick question in this regard? That can this be then carried to some industry, let's say pharmaceutical industry, and at least have one example of something that is discovered as a result of this database and which is really important?
Yes, so there are a couple of issues. So the Indian biodiversity law doesn't allow any traditional knowledge to be patented. So I am pretty sure, and I don't want to name anything on record, people in US and top-notch pharmaceutical industry are using our database. But they will not acknowledge it. Because if they say they got the information from traditional knowledge, they cannot patent it. Did you understand? So anything which is coming out of traditional cannot be patented. And the drug discovery industry wants the molecule or whatever active ingredient to be patented. This can certainly be used for sort of drug discovery. And the very simple analysis, as I said, what is common and what is unique can lead to very important new hypotheses.
Thank you.
So, yeah, can we move on? And there is one more question from Professor Alakre. He has already typed, so let me read for him. Once a compound is postulated to be a candidate drug for the amelioration of a disease in the Western medical system, it has to go through a set of clinical trials. Could you give us examples of characterization of phytochemical of the medicinal plant which has gone through a clinical trial either in the West or in India?
So I had the example in the second slide. So of course the artemisinin is a good example, and paclitaxel. And actually artemisinin we have now engineered E. coli. I mean, people have engineered E. coli to produce artemisinin. So these are good examples of things which are past clinical trials. And there are of course several natural products, as I showed in that slide 21. Sorry, here, which have passed the US FDA drug space, approved drug space. So natural products still continue to contribute quite a lot to drug discovery. Did I answer the question?
Yeah, I hope so.
Yes, you did. I mean, it was somewhat similar to what Vivek was asking, but yes, you did. I missed part of your talk, and that's why I didn't know. Thanks.
Okay.
So the I can point to phytochemicals which have passed clinical trials. So there are clinical trials which are at two levels. They could do it at the level of plant extracts, okay, or they could do the chemical, the active ingredient part. So I gave you examples of actually active ingredients which have passed through that.
Okay. I don't see any more questions. So just a minute. Yeah. So therefore, just let me see if yeah. Okay. No other questions. So thank you very much, Areejit, for your very nice talk, and also nice discussion that followed the talk. Also thanks to Professor Vivek Datar, who has actually proposed this talk. Thank you both of you. Thank you also all the audience, both from the TIFR family and outside, who participated and made this talk much more enjoyable. Thank you very much.
Thank you very much. I really appreciate the kind words. Thank you.
Thanks. Yeah.